Welcome, everyone. My name is Ulrich Broeckel. I'm from the section of Genomic Pediatrics at the Department of Pediatrics here at the Medical College of Wisconsin and Children's Hospital of Wisconsin. And I want to thank Bionano for inviting me to give a presentation today about optical genome mapping for constitutional chromosome analysis. I just want to give you a brief background of on our lab and the interest we have in optical genome mapping. We participated in Bionano's postnatal study to evaluate the performance of optical genome mapping for constitutional chromosomal abnormalities, and it's my great pleasure that on behalf of the whole group, I can actually present some of the results from this manuscript, which was just recently published beginning in January in the Journal of Molecular Diagnostics. I also want to give you a brief overview of our ongoing prospective study to recruit patients here at our institution, where we combine arrays, exome, whole genome sequencing, and optical genome mapping. And we're in the process of establishing optical genome mapping for chromosomal abnormality testing as a clinical test. Just to recap, you might be aware of this already, a platform comparison between optical genome mapping and current other cytogenetic methodologies, such as karyotyping, FISH, arrays, and as well as next generation sequencing. And next generation sequencing really here mainly focuses on short read sequencing. So this highlights in green, yellow, and red the variations, abnormalities we can detect with the various technologies. And for example, on karyotyping, the resolution in terms of the size, what we can detect, for example, on karyotyping deletions in a megabase range of about 5 to 10 megabases. Obviously, karyotyping also allows you to detect duplication, translocations, and inversions. Same for FISH. When we look at this very targeted, again, we're all aware that FISH is a targeted approach. Arrays, for example, the limitations obviously well established that we cannot detect translocation and inversions, although we can detect deletions and duplications with a much higher resolution. Similar to next generation sequencing, the limitations are for inversions, potentially translocations. And if you look at optical genome mapping, and this is really one of the reasons why my lab and we got interested in this technical approach, is the expanded ability in one assay to detect at a very high resolution, really the broad spectrum of clinically relevant structural variations and abnormalities, which this technology allows us to detect. Again, here, just to summarize this, we can detect structural variation in a range of about 500 base pairs to a megabase. We can detect structural variants in mosaic samples up to roughly about 5% with a sensitivity and false positivity, which is in the range for a diagnostic test. And here you can see the different abnormalities we can detect. So I want to present a few examples, a few results from this study, which really aim to evaluate optical genome mapping and compare it against standard of care for postnatal constitutional disorders here. And the goal of the study, and as I said, was just published in the Journal of Molecular Diagnostics, went online in January. The goal was to evaluate OGM and compare this with the performance for standard of care. The study design was that we had 9 study centers, which all contributed samples and the analysis. The review included a large number of retrospective samples, as well as a prospective sample collection. And mainly the cases include neurodevelopmental disorders, autism spectrum disorder individuals, intellectual disability, development disorders, and ADHD. We include a small set of families. All the laboratory tests were performed in the procedures were performed in the different diagnostic labs or in the different labs. Repeated multiple labs, and we had a very standardized data analysis with a multi-investigator, multi-level review of the results. Here, just the sample characteristics briefly. You can see the distribution of the samples we included, and here on the the right side, the available results for the comparison to testing with arrays, karyotyping, and FISH. Some of the samples, as you can see, also had multiple tests performed. So just to highlight, obviously one of the main questions is the concordance between those. And as you can see here, the concordance rate was extremely high, over 98%. For these for all these samples here. The other question was, or question you could ask is like, so how many cases were recategorized based on the OGM results? We recategorized those based on the highest classified variant found according to ACMG guidelines, and the changes between the standard of care and OGM were really predominantly due to obviously new findings, which were either undetectable by the transitional cytogenetic methodologies, which were used for these particular samples, or also new clinical information about genes and loci since the sample was originally analyzed. And as you can see here, there are a few samples which were recategorized, which were negative before, which were recategorized to a variance of unknown significance. The samples of likely pathogenic to pathogenic, a few were recategorized to variants of unknown significance. So you can see here the results for OGM and how this was recategorized. The other thing which was is probably also really interesting that I would like to mention are the diagnostic results. So if we look at the retrospective study, the standard of care, which is focused on pathogenic or likely pathogenic, we had about a rate of 17% with OGM that increased slightly to 22.5%. If we look at all reportable ones, that increased from 48 to about 54. Interestingly, if we focus on, and that it's a smaller number of samples, obviously, but in the prospective study, we had about 5% likely pathogenic and pathogenic, and that increased to about 10%. And overall, the total reportable results were with standard of care about 28%, and it increased with OGM to about 40%. So a quite noticeable increase in the total reportable results we saw in these samples. So I want to review with you a few cases here just to highlight also some of the platforms. And this is really very much work in progress. We're, as I said, we're trying to combine different technologies and approaches for cytogenetic testing. And so I want to showcase that on a few cases here. So this is a case included in our prospective study of a 4-year-old female. You can read the clinical presentation, microcephaly. And so as part of the study, we performed an array CGH, optical genome mapping, as well as also whole genome sequencing. This is not done as a routine workup. This is really part of our clinical study here. So I want to show you the results, typical result where we detect using an array, and that would be cytoscan. And you can see here, obviously, it looks like a translocation. You have an increase here and you have a deletion here. And so it looks very much like this could be a translocation, but obviously, arrays are not able to detect that. And so we performed optical genome mapping. We found a number of different variants, but we see the loss in chromosome 15 and findings in chromosome 2 and chromosome 15 very much suggest that we have here interchromosomal translocation, as you can see here, noted by the line between these two chromosomes. And if we look at the next result, you can clearly see here the complex rearrangement, what we see here, the gain on chromosome 2, the loss on chromosome 15, and the chromosome 2 to 5 translocation. So very straightforward to detect this in one single test. The other thing what we did, and I told you that we also performed whole genome sequencing on this particular sample. And so this is something we started to work right now, just initiated to work with Bionano using their new VIA software. That is a software which combines multiple platforms, the results from multiple platforms, like, for example, next generation sequencing, microarray, and optical genome mapping. It uses this to combine it and visualizes that, and also provides annotation tools. And really, the underlying concept of this is that, by combining the analyses for from these different platforms, that this really increases the efficiency for reporting. And so we just started to work with this. So this is very preliminary here right now, but this is the result if we combine all this for this particular case with optical genome mapping, array, and whole genome sequencing. Obviously, we can detect these the duplication and deletion with arrays as well as whole genome sequencing, very straightforward. The translocation then can be detected with optical genome mapping. So I think we're going to expand on this and use that. So we're really just in earlier stages, but I think this provides a very nice platform to combine various tests. In particular, if we're thinking about combining next generation sequencing results with optical genome mapping, and I think that provides quite an opportunity for the future to develop new analyses approaches. So the other thing what we're doing, and so the results what I showed you, the whole genome sequencing, this was short read. We also started to work and explore what it takes to combine OGM, optical genome mapping, with long read sequencing. And the platform we're currently exploring right now would be Oxford Nanopore technology, nanopore sequencing as a methodology for long read sequencing. And it is very likely that compared to short read sequencing in the constitutional space, long read sequencing will have, or does have, certain advantages, and that with continued development and improvement of the methodologies and analysis approaches, that this is a very promising technology. It has advantages over short read and in terms of for structural variation, as well as for regions which are obviously difficult to assembly, like when we're looking at pseudogenes, regions with high homology, or repetitive regions. And again, as I said, the potential for a combined analysis to increase the efficiency is obviously pretty apparent. So I want to show you some very preliminary results to showcase what we see with Oxford long read sequencing here. And one of the things which is probably worth mentioning, at least in our experience, that the software to analyze larger structural variation with Oxford with long read sequencing is not as mature. If we're looking at regions of about 300 kb to up to 4 megabases, we see a lot of signals for structural variation, but it's really not currently assembled in a way that this represents the finding here. But what I want to show you is one case where we've seen here on the lower side, we see the signal with optical genome mapping, which characterizes a region in a particular in a gene ELP2, which we thought could potentially be relevant for the phenotype. And when we did long read sequencing in this case, we can really clearly pinpoint two potential breakpoints here. Obviously, sequencing in this case has the higher resolution when we're looking down at particular genes or coding sequences to really identify this, as you can see here, compared to optical genome mapping, given the density of the probe. So this is where we think the combined approach really allows us to zoom in and detect and then further characterize this. I think we also see this as obviously as an opportunity to define breakpoints more clearly. So next steps, obviously, what we're looking at is we have interest in and we're planning on developing optical genome mapping as a clinical test. We're in the process on finishing up our validation. At the same time, we're all aware that insurance billing and the billing framework needs to be developed, and so we're pursuing this as well. And as I think I could show you here, we truly believe that a multi-platform analysis is likely to gain substantial interest, and we're looking at optical genome mapping with short read sequencing. Long read sequencing are certainly methodologies which are currently being developed. They're going to expand in their use. And the technology will improve both on the just sequencing and accuracy side, as well as the analysis methodologies, which in our experience still will need to significantly be developed and improved. But we clearly see that the potential of combining this having a platform and exploring what a platform can do, which can visualize multiple analysis methodologies. I think we see this as a great potential. And we also see potential in really, for example, hybrid assemblies where we have OGM-guided sequence assemblies for certain regions. I think there's potential here as well. And again, the methodology for that, the methodologies need to be developed. So with that, I want to acknowledge all the people who contribute to all this and make this work possible. First of all, people at Bionano Genomics. I want to thank all the investigators and who were involved in the postnatal study, which was supported by Bionano Genomics. And finally, I want to thank my team members here, people in my lab at the Medical College of Wisconsin, as well as in our genetics clinic, the genetic counselors and the members in the clinic as well. With that, again, thank you very much for your attention, and I'll hand it back.